Welcome to video 7 of Genetics the Code Broken. In this video, we're going to be looking at recombinant DNA, the technologies which we use to produce it, and then how we can use this recombinant DNA in gene mapping. So recombinant DNA is naturally produced in cells as a result of chromosomes crossing over in meiosis, but it can also be produced in the laboratory using molecular technologies. So it is where two different sequences of DNA have, of those chromosomes have combined to produce a new sequence. So one of the ways we can produce it is by using restriction enzymes, which we'll come to in one of the future slides. So what are some of the technologies which we use to produce and then use recombinant DNA? So the first is when we cut and rejoin DNA using restriction enzymes. So this allows us to cut pieces of DNA and then seal them back together using enzymes or ligases, and it means that the DNA strands have now been attached at their base pairs, and we now have a new and different sequence of DNA. We can also use vectors, such as bacterial plasmids, which act as carriers of DNA fragments or genes. It allows us to insert different sequences of DNA. PCR, or polymerase chain reaction, is a technology which is used to produce multiple copies of a DNA fragment. DNA hybridization allows us to compare similarities in gene structure and therefore understand where these sequences may have come from. We've got gel electrophoresis, which allows us to sort fragments of DNA according to their length. And then southern blotting is another technology which we can use, which allows us to take these DNA fragments that have been separated using electrophoresis and then do some future testing on them on a more solid surface. So once we produce this recombinant DNA, we can also use this recombinant DNA technology to map genes. So by being able to identify genetic markers, it means that we could use these markers to recognize offspring. The use of these markers also allows us to determine whether genes are linked and also how far the distance is between some specific genes. The main techniques which we'd use to identify a sequence of genes and their positions are restriction mapping and DNA sequencing. So the first is restriction mapping, and that's again where we use a restriction enzyme to cut pieces of DNA, but we know where this restriction enzyme is going to cut, and we term this the restriction enzyme site. Once we cut this DNA up, many copies of this fragment are produced using PCR. We can then label these fragments using radioactive phosphorus, and when some samples are cut by either one or two different restriction enzymes, we're able to determine the size of the DNA fragments using electrophoresis, and therefore we're able to deduce their position on the original fragment. So here we have a DNA fragment which has been cloned by the PCR technology and labeled with a radioactive phosphorus, and we know that it's 1,000 base pairs. We then use another restriction enzyme. We can see that they've used both 1 and 2 and both restriction enzymes to produce fragments by different sizes, and it allows us to see those because of that radioactive phosphorus. And then once we know that, we can use these 3 separate electrophoresis results to deduce the site of these restriction enzymes in the original fragment, and thereby working out the different sizes of each DNA sequence. Second is DNA sequencing. So it's where we can identify DNA sequences on a chromosome using genetic probes in hybridization. So one of the main examples is FISH, or fluorescent in situ hybridization, and it's where we use a probe, and a probe is a specific sequence of DNA which is complementary to the DNA bases on a gene or part of a gene. And then once we have produced these probes are mixed with DNA samples, and it's heated so that the DNA denatures, and therefore we produce single strands. And then when we cool it down, the probes are allowed to bond with single strands of the DNA, and then this will cause this probe to fluoresce under a fluorescent light, and thereby deducing where the position of the gene is, and then that can be recorded down. Therefore, we have located the position of that gene on a chromosome. This is mainly used in genetic screening for abnormal chromosomes and defective genes. So here we have two kind of images. On the left, we have the process in which recombinant DNA is produced using a plasmid as a vector. So we can see that the plasmid has to be removed from a bacterium, that we cut both the gene and the plasmid using the same restriction enzyme, and then that gene that we've cut from the chromosome is added to the plasmid using a DNA ligase. And on the right, we have a picture of how the fish process works. And we can see that we finally see that the chromosome is fluorescing in the position as to where that probe has hybridized with that section of DNA. Thank you for watching video 7. Make sure you tune in for video 8.